It's incredibly easy to picture a cell as just, well, a tiny static water balloon floating around under a microscope, right? But when we actually peek inside, oh man, that image couldn't be further from the truth. What we're really looking at is a bustling, highly structured microscopic city. We're talking highways, storage warehouses, and even, get this, quality control shredders. Welcome to this explainer on the anatomy of a cell. Today, we're going completely microscopic to uncover exactly how our fundamental biological building blocks organize themselves to, well, keep us alive.
Okay, let's dive into this and map out our journey. Stop 1, the internal cellular medium. Stop 2, inclusions and storage. Stop 3, protein quality control. Stop 4, the framework, or cytoskeleton. And finally, stop 5, filaments and cellular motors.
So kicking things off with part 1, the internal cellular medium.
Before we can start building the structures of our microscopic city, we really need to understand the environment they sit in. Now, there's a super common misconception we need to clear up right out of the gate. People often use the words cytosol and cytoplasm interchangeably, but they are definitely not the same thing. The cytoplasm is basically everything from the nucleus all the way out to the cellular membrane. It includes all those tiny organs, the organelles. But the cytosol, that is the true internal aqueous medium. It's this watery matrix made up of roughly 70 to 85% water. Think of it kind of like the unstructured broth of a soup, where countless enzymes, signal molecules, and building blocks are just dissolved and floating around.
And just to give you an idea of the sheer massive scale of this fluid, the cytosol takes up, on average, 50% of the entire cell's volume. Literally half of the cell is simply this concentrated, unstructured, watery base. It fills up every single microscopic gap that isn't already occupied by mitochondria or other structured elements. It's a whole lot of liquid.
Moving right along to part 2, inclusions and storage.
Floating around inside all that liquid broth, you'll find these distinct structures that completely lack a membrane. These are basically our cell's storage warehouses. And this brilliantly illustrates what I mean. Just look at the enormous balloon-like empty spaces in these adipocytes, which you probably know better as fat cells. Those are massive fat droplets. The cell has hoarded so much energy reserve here that this single inclusion droplet literally pushes all the other cellular machinery right to the absolute edge. It occupies almost the entire cytosol.
But hey, not all storage is about hoarding future energy. Sometimes it's just about holding on to waste. As cells like our neurons or heart muscles age, they start to accumulate this yellowish-brown pigment called lipofuscin. This is essentially the cellular aging pigment, created by a buildup of oxidized lipids and proteins over an entire lifetime. You know those age spots that start to appear on human hands as we get older? That is the real-world visible proof of this microscopic accumulation happening right inside the cells of your skin. It's pretty wild to think about.
All right, let's look at part 3, protein quality control.
Now that we've seen the warehouses, let's take a look at the actual workers. Out here in the fluid, there's this dramatic narrative playing out every single second between the proteins that carefully build and the industrial machines specifically designed to destroy. On one side, we have the chaperones. Proteins actually need to fold into highly specific 3D shapes to function properly, but they don't always get it right on their own. Enter the chaperones. They act as the quality control inspectors, providing the perfect isolated environment and exact timing to ensure these proteins fold perfectly. But what happens when a protein is damaged or just gets it totally wrong? Well, that's where the proteasomes step in. These are menacing, cylindrical enzyme complexes that act, literally, like cellular shredders. Their entire job, to destroy damaged or useless proteins.
So how does this cellular hit list process actually go down? It happens in 3 crisp steps. First, bad protein folding occurs, or maybe a protein just gets damaged. Second, the cell identifies the target and chemically marks it by attaching tiny polypeptides called ubiquitin. That ubiquitin is essentially a giant bullseye painted right on the protein's back. Finally, step 3. The marked protein is dragged over and mercilessly shredded inside that proteasome cylinder. If you follow the visual flow of this cycle, you can notice how the target protein winds its way along. Once it gets that ubiquitin tag, man, it is a completely one-way trip. It gets fed directly into the hollow center of that proteasome cylinder, and out the other end pop completely harmless, broken down amino acids, totally ready to be recycled. It's just an incredibly efficient, ruthless system. Let's move to and see how this builds in part 4, the framework or cytoskeleton.
So, we've talked about the fluid, the storage, and the quality control.
Now we need to look at what physically holds this entire microscopic city together. The cytoskeleton can seem overwhelmingly complex at first glance, but we can actually neatly categorize it into two main components. First up, the structural scaffolding. These are the filaments. They come in three specific types: microtubules, actin, and intermediate filaments. Their whole job is to provide structural shape, anchor things into position, and allow for motility. The second category consists of the accessory proteins. Think of these guys as the operational workforce, the regulatory, linker, and motor proteins. They control how the filaments grow, connect the different parts together, and physically move organelles around the city.
Let's briefly highlight the middle child of this scaffolding system, the intermediate filaments. They sit right at 10 nanometers in diameter. So exactly as the name suggests, they're thicker than the delicate actin threads, but thinner than those massive microtubules. There are a few types of these, 6 actually, like keratin, which you definitely recognize from your hair and nails, and neurofilaments found in your brain. They act a lot like internal steel cables, providing absolutely vital tensile strength across a bunch of different cell types.
And finally, bringing us to part 5, filaments and cellular motors. For our final stop, we're really going to ramp up the energy, because a city isn't just about static scaffolding, right? To have movement, you need roads. Enter the actin filaments. These are incredibly thin, flexible, braided chains made up of globular actin molecules. They constantly crisscross right beneath the cellular membrane and span the entire cell. Basically, they create the main pathways for continuous internal movement and handle massive tasks, like the physical contraction of your muscles.
So the crucial point is, a road is completely useless without vehicles. And that's exactly what motor proteins like myosins are. They generate intense traction forces, but hey, they don't do it for free. They literally consume ATP, which is the cell's main energetic currency, as fuel. With that energy, these myosin motor proteins act like tiny, purposeful delivery trucks. They physically step along the actin highways to drag entire cellular structures from one side of the city to the other, or to pull filaments together to make your muscles contract. It is truly mind-blowing.
We've seen today how beautifully organized and bustling a single cell is, from its fluid highways to its ruthless recycling plants. But I want to leave you with this thought. If our cellular cleaning shredders and our internal scaffolding dictate our physical strength, our movement, and even how our skin and organs age, what exactly happens to our bodies when these microscopic systems inevitably begin to slow down and misfire? It's a fascinating question, and one that gets right to the very core of human biology. Thanks so much for joining me on this explainer, and keep exploring.